for example, in a cartoon form. This is the first evidence for lymphatics in the intracranial structures. Also, we learned about a lymphatic or fluid-flowing system that percolates through the interstitial space that, for example, is clearing things like beta amyloid and unwanted metabolites. These are all new concepts, but they deal mainly with the meninges and the interaction with tissues below. Fanny Harrison in my laboratory was interested in what about the interaction between the meninges and the tissues that sit above it. And so in this paper in Nature Neuroscience, published in November of this year, she identified for the first time direct vascular channels that connect skull bone marrow and the brain surface, enabling myeloid or white cell migration. I'm not going to tell you how she went about it, but I want to show you what the channels look like. So you can imagine, this is work done using confocal microscopy. You can imagine that this is the bone in the marrow cavity here, and you can see these tubular extensions. Just to give you a frame of reference, the blue is bone, the red are blood vessels. You can see them coming down to these channels. These are the channels. And they're moving in towards the dural surface, and based on, actually there were 4 publications that came out since November of 2018. So this is a very hot topic, and all of them came in Nature publications. Fanny validated this concept of microchannels using micro-CT. Here is shown in a mouse, and you can see this is the bone proper with the marrow inside, and these bone-protected tubules which contain blood vessels. So the meninges are constantly talking back and forth with the bone marrow, which we think is really quite important in migraine. And there is one disease that I just have a brief period of time to talk about, and it's completely unrelated to migraine in any way, but it tells you fundamental biology that deals with these microchannels and how important they can be in disease. In this case, it's acute lymphocytic leukemia. And this cartoon shows very nicely the anatomy, as I was mentioning before, with the bone, the dura, the CSF space, and the arachnoid, the pia mater, and the brain. And what this group from Duke was interested in understanding was why the high occurrence rate of CNS complications in leukemia. And in particular, they were interested in why the meninges, as opposed to parenchyma, was such a target for acute lymphocytic leukemia, unlike other kinds of tumors which invade the brain parenchyma. And they were able to answer it, and answered it in a beautiful, elegant way that I think I can describe in 2 minutes. They labeled leukemic cells with fluorescent markers, and then inject them in the tail vein. And they looked in the brain parenchyma, and they did see, just like in cancer cells, these leukemic cells attach to branch points and blood vessels. But unlike cancer cells, these cells can't penetrate the blood-brain barrier, okay? But they get into the meninges, and how they do it is by getting into the bone marrow. And in the bone marrow, rather than crossing into the blood vessel to get into the brain, they actually crawl on the outside. And they were able to show this in a very elegant way, identify what the receptor interaction mechanism was with the blood vessel and these leukemic cells. And using neutralizing antibodies, they were able to block the development of CSF invasion by these leukemic cells, and they did it with a small molecule as well. It's a very beautiful, elegant story, very clinically relevant. But it points out the fact that there is the ability of cells, in this case neoplastic cells, to get into the CSF. They don't have to cross the blood-brain barrier to do it, okay? And the same thing is true in those channels that Fanny was described. And this would be the equivalent of the channel. You don't need to cross the blood-brain barrier. And that's why under conditions of stroke and inflammation in the brain, the skull donates the cells that are the white cells that are first responders, okay? Because they don't have to break down the blood-brain barrier. They get right into the central nervous system, which I think is a very important concept. So, how does migraine relate to it? Well, there's a lot of crosstalk that's going on here between the brain, and you know there's signal migration. Cortical spreading depression is occurring in the brain proper, but yet the dura mater is showing the inflammation as well as some elements of that molecular cascade that Dahlker described. But there's migration of the signal. How does that signal arrive in the dura mater? We're suggesting that these microchannels might well be part of the story, and the interaction with the bone marrow and the meninges might well be another part of it. So, to summarize, 40 years of work, and my former students have done it elegantly. The meninges contain these afferents that are implicated in headache. They're neuropeptide containing. They're released from afferent and central trigeminal vascular terminals. We know about the drugs, and they're targeting the peptides and the serotonin receptors. We know that cortical spreading depression underlies the aura and is an upstream driver in a migraine aura attack. It upregulates inflammatory signaling in the brain and the meninges. We saw evidence for that. And then this last part of the story, which I want to leave you with, because 40 years, that's the history. We want to look forward. That's the most important thing. The society needs to be forward-looking. What's going to be the next treatment down the line? What's going to be the mechanisms that are going to get us to treatment? Will this provide a new template, what I've been talking to you about? I think only time and additional experiments can tell. But I also want to point out that that mechanism that I described with the microchannels, I believe will be quite relevant for other disorders affecting cortical ribbon, such as brain trauma. And this federation and docs really do treat a lot of brain trauma. The infection, MS, I mentioned leukemia, and I would put a bet, it's probably migraine as well. So, thank you so much. It's really been totally wonderful. Thank you to my students, to Lars. Thank you to the chairman. And have a wonderful evening. Thank you for listening.